In this session, let's say a few more points on the heart of human, and then later we'll also see the transverse section of the blood vessels, and then we will also see about the cardiac cycle. So now first let's begin with this. A few more points on the structure of the heart. So here I would like to talk about some special nodal tissues which we call them SA node, that stands for sino-auricular node, and another one called AV node, and this stands for atrioventricular node. So we'll take right. So here we already know. So these are the valves which are present, the bicuspid and the tricuspid valves, and these are the openings actually, the atrioventricular apertures. So here in the right atrium, so we know this is the right atrium, and this is the left atrium, and this is the right ventricle, and this is the left ventricle. Okay, so here in the right atrium near the opening of superior vena cava, so which we have seen in the last session, here we have a small group of cells. We call it a nodal tissue, and this is what I call it the SAN, that stands for the sino-auricular node, and one more is present close to the atrioventricular septum, and this is called the AVN, the atrioventricular node. So both are the nodal tissues, and here this sino-auricular node has, as I told, it has a nodal tissue and it is auto-excitable. It is auto excitable, means capable of getting excited by itself. So it produces at regular intervals uh, impulses that actually help in contraction of the heart, right? So that is here, I'll say, at regular intervals it generates action potentials. So each action potential corresponds to a contraction, and that will be followed by relaxation. So here, okay, so we have two right nodal tissues, both located in the right atrium. One is present near to the opening of a superior vena cava, and another one is present very close to the atrioventricular septum, and these are connected by the fibers, nodal fibers. Then arising from this AVN, atrioventricular node, is atrioventricular bundle. So it is called atrioventricular bundle, and this atrioventricular bundle, okay, comes up to uh, right into this interventricular septum. So on to the top of this interventricular septum, so here this atrioventricular bundle, it starts dividing into two branches. So it divides into two branches, and one of the branch enters into the wall of left ventricle, and another branch enters into the wall of the right ventricle. So this is we call the left bundle, and this one is called the right bundle. So this left bundle, after entering into the wall of the left ventricle, it starts, okay, branching off. So these are called the Purkinje fibers. So which also is seen on the right side on in the same way, and here these are also the Purkinje fibers. So here this left bundle plus the right bundle plus, okay, the Purkinje fibers. Okay, so here the left bundle plus the right bundle and the Purkinje fibers all together will result in the formation of uh, something called bundle of HIS. So bundle of His. So this is named after a person. So this bundle of His is named after a person, okay, whose name is Wilhelm His. Okay, and he is the person who discovered this bundle of his, whatever this left bundle and right bundle and the Purkinje fibers. So in 1893, okay, named after a person Wilhelm His, right? So here we understand that SAN node and the AVN node are both present in the right atrium and connected by the nodal fibers. Okay, so this SAN is auto excitable and it generates action potentials at regular intervals. So for this reason, the SA node is what we call the pacemaker. So it is the only one, the pacemaker, present in the human heart. So if the question comes that the number of pacemakers in human heart is only one, so only this SA node is called the pacemaker. Whereas, right, this AV node, we have another name. We call it node of Tawara. So we call it node of Tawara, right? So here coming back to this SAN node, right? The number of action potentials generated by SAN, that is sino-auricular node, is around 70 to 75, right, action potentials per minute. So it will be 70 to 75 action potentials per minute. As I already told you that each action potential corresponds to a heartbeat. So hence here you can take this action potentials average as 72, okay, so beats per minute. As I already told you that each action potential corresponds to a contraction. So here I'm taking, okay, an average of 72 beats per minute. Right, so here one more point. So these are can take as internodal pathways. Right, so that's all about a few more points about the heart. So again, when I come to the cardiac cycle, we will again discuss about these points: sino-auricular node, auriculo-ventricular node, and how actually they help in contraction of the heart and pumping the blood to all parts of the body, and again collecting back into the heart. Okay, so now let's see the transverse section of the blood vessels. So tiers of artery and vein. We'll also see about the capillaries also in transverse section. So what is the difference between the wall of the artery, the wall of the vein, and also the wall of the capillaries? So if we see from arteries to capillaries, the diameter decreases from arteries to capillaries. So already I have mentioned, so in the earlier sessions, that arteries have a bigger diameter and they divide into smaller branches, what we call them arterioles, and each arteriole again further divides into much more smaller branches, what we call them the capillaries. So this is artery, and this is arteriole, and this is a capillary. And we know that all these capillaries will reunite to form a venule, and other venules unite to form a vein. Now we'll see the transverse section. So let's see here, if I take a cut on this artery, how it looks, and then we'll see even the transverse section of the capillary. And we already know that these will unite to form a venule. So this is a venule, and then this is a vein. Even we'll see the transverse section of a vein. Now in transverse section, okay, both arteries and veins have, right, the following layers. They are: one, tunica externa; another one, tunica media; and another one, tunica intima. You can also call this tunica externa as adventitia, and tunica intima also as interna. I'll write like this. So tunica interna or intima. So total we have three layers: tunica externa or adventitia, tunica media, and tunica interna. So right, I have drawn the diagrams here. We'll see this is the tunica externa, and this one is tunica media, and this is the innermost layer, that is tunica interna. So this is the outer layer, and this is the middle layer, whereas tunica interna is inner layer. Of course, we can easily understand with the names itself, the externa, media, and interna. right? So here this is the transverse section of vein, and this is the transverse section of artery. So in this case also you will see same, this is tunica externa, and this is tunica media, and this is tunica interna. The tunica externa is composed of fibrous connective tissue and also collagen fibers. So this is composed of fibrous connective tissue, plus we will also have these collagen fibers are also present, whereas the tunica media is the one that consists of smooth muscles. So the media consists of smooth muscles, and it also has elastic fibers, and coming to the tunica interna or intima, so this consists of squamous endothelium. So it consists of a squamous endothelium layer. So this is what each layer is composed of. Okay, so composed of fibrous connective tissue plus collagen fibers. Okay, then the tunica media consists of smooth muscles and elastic fibers that help in the smooth muscles help in contraction and relaxation of the blood vessels. Of course, that takes place okay autonomously and are under control of autonomic nervous system, and the elastic fibers may help in giving elasticity to some extent okay for these blood vessels, right? So here when coming to this smooth muscle layer, so this smooth muscle layer is much thicker in arteries. So I'll mention here the muscle layer is thicker in arteries. Here the problem is in case of arteries there will be a lot of pressure. So for that reason you can see the thickness of this artery wall is okay very heavy, and the okay the artery I mean sorry the vein wall in this case is thinner when compared with the artery and vein, and you can also see the lumen is narrow. So this is the lumen of artery, and when compared with the artery the vein have okay a bigger lumen. So you can see here the difference in the thickness of the wall. So I said that artery has okay thicker muscular layer that helps in giving okay extra strength to this, because the arteries will have more pressure inside than the vein. And again one more point is these veins and also the lymph vessels have valves. The veins and the lymph vessels will have valves, whereas arteries do not have valves. These valves help in flow of blood in a particular direction, that is mostly towards the heart, right? So arteries do not have the valves, whereas veins and the lymph vessels will be having valves, right? So again you can also see the tunica media. In case of the vein it is comparatively thin when compared with the artery. You can see the tunica media here, it's okay bigger, and in this case I mean say it is thicker, and in this case the tunica media is thinner. So you can see the tunica media is thinner in vein when compared with the artery. Okay, fine. That's a small information on transverse section of artery and veins. Now, let us see the even the transverse section of capillaries. So how the capillary wall is different from these veins and arteries. Right, we'll see about transverse section of capillary. So the capillary wall is very thin because it has to allow the movement of different molecules between the ECF and the blood that flowing inside these capillaries. So when you take the transverse section of the capillary, you will see an outer basement membrane, and then below that you will find only one layer, that is endothelium. So you can see in this case the endothelium is the tunica interna. Okay, artery and vein will have this endothelium, that is the innermost layer. In this case of capillary, you don't find this externa and media. There is only, okay, the inner layer, that is endothelium. Only one layer, okay, that is squamous endothelium is present. So this is the basement membrane, and this is the squamous endothelium layer that is seen in the case of the capillary transverse section of capillary wall. So here this is the lumen where the blood flow is seen. Right, so here you can see very clearly the difference between the capillary and the vein and the artery differences we need to know. So in what regard these all differ from one another. Okay, so the tunica media is very thicker in artery, whereas it is thin in case of media. That is what I just mentioned, that muscular layer is thick in case of arteries to withstand the pressure that will be that is actually will be seen in case of arteries. Okay, in capillaries you don't find any of these layers. You'll find a basement membrane and one layer of squamous epithelium. This allows the movement of okay, or the diffusion of gases, the oxygen and carbon dioxide, and many other molecules like glucose and other nutrients that will be freely moving, okay, right through this squamous endothelium from in and out of the capillary. Right, this is the difference between the TS of capillary, the artery, and the vein. So now that's all about these blood vessels. Now let's see about cardiac cycle. So here the contractions and relaxations of heart that are cyclically repeated is what we call the cardiac cycle. So the contractions here are called systole, whereas the relaxations are called diastole. So systole and diastole. Systole, which means the contraction of the heart, and relaxation, which means the diastole. So just now we have seen about the AV node and also SAN node, and I told that SAN node is the pacemaker, and it is the point from where the action potentials actually start. We'll see here. right? So I told here near the opening of the superior vena cava we have SAN node, that is auricular node, and okay, near the base of these ventricles, or okay, near the interventricular septum, we have one more nodal tissue, and this we call it AVN, okay, atrioventricular node. And from here we know atrioventricular bundle, and that bundle will divide into left bundle and right bundle. The left bundle and the right bundle will enter into, okay, the left and right walls of the ventricles, and they form Purkinje fibers, and this is atrioventricular bundle, okay, and this is the left bundle, and this one is the right bundle, okay, and these are the internodal pathways. Okay, so we'll see, as I told, that SAN node is the pacemaker, and it is the point from where the action potentials generate. So those action potentials will spread all through this all through the walls of auricles, which results in the contraction, and then the action potentials finally reach the atrio ventricular node. So from the atrioventricular node, the action potential passes through the left and right bundles and then to the Purkinje fibers, and this results in the contraction of the ventricles. So here first the contraction of auricles and then the contraction of, okay, ventricles, and this all is initiated by the pacemaker, that is SAN, that is sino-auricular node. So now let us see those steps in the, okay, heart beat, that contractions and relaxations. So I'm going to put them in three main steps. The first one, atrial systole, and second one, ventricular systole, and third one I'll just mention as atrioventricular diastole. So total we have, okay, so atrial systole, where ventricular, okay, systole, and atrioventricular diastole. What did I mention? The systole, which means contraction, and diastole, which means relaxation. So here, so each heartbeat lasts for 0.8 seconds. So each heartbeat lasts for 0.8 seconds, and I'm going to divide that 0.8 seconds, okay, for this atrial systole and ventricular systole and atrioventricular diastole. So this atrial systole will be around 0.1 second, whereas the ventricular systole is around 0.3 second, whereas atrioventricular diastole, that lasts for 0.4 seconds, which altogether will be 0.8 seconds, right? So you'll see here, by the time the pacemaker initiates this atrial systole, okay, so this is the left atrium and this is the right atrium. We know that the right side will be filled with deoxygenated blood that will be brought by the superior vena cava, inferior vena cava, and also the coronary sinus that brings the deoxygenated blood from the wall of the heart. And okay, this right atrium will be filled with the deoxygenated blood, whereas the left atrium will be filled with oxygenated blood that comes from the lungs through the pulmonary vein. And now okay, so when this pacemaker initiates an action potential, okay, this will, okay, will spread all over the atrium, and that results in atrial systole, and this will result in pumping the deoxygenated blood from the right atrium into the right ventricle. And okay, at the same time, from the the oxygenated blood from the left atrium is pumped into the left ventricle, right? So when the SAN node initiates an action potential that spreads through the walls of auricles, and that results in the contraction of the auricles, that's what we call the atrial systole, and this will, okay, pump the blood, that is deoxygenated blood present in the right atrium, into the right ventricle, and at the same time the oxygenated blood present in the left atrium is pumped into the left ventricle. So that will be around 0.1 seconds. The next one, right, the ventricular, right, systole. So that the atrial systole is followed by the ventricular systole, and when the ventricular systole is taking place, okay, so by the time the action potential reaches to the AVN node, and that passes through this, okay, left bundle, right bundle, and then finally to Purkinje fibers, and this results in the contraction of ventricles. And when the ventricles contract, there is a chance of the blood that is present in the right ventricle to get back to the right atrium. At the same time, okay, the blood from the left ventricle to get back to the left auricle. But this will not happen, as there are the valves, the tricuspid valve guarding the right side, and the bicuspid valve, okay, guarding on the left side. They close, preventing the backward flow of blood. Okay, so during the ventricular systole, what happens is these valves will close, preventing the backward flow, and the contraction, right, I'm sorry, the closing of tricuspid and bicuspid valves produce, okay, the heart's first sound. So first heart sound. So this first heart sound is what we call it the lect. Clear? So closing of tricuspid and bicuspid valves produce the first heart sound, that is lect. So the first heart sound is lect. Now the blood, as it cannot pass back into the respective auricles, there is only one way that we know. From the left ventricle, aorta arises, and from the right ventricle we have a pulmonary artery. Okay, here I'll show it. So from here is the aorta from the left ventricle, and from the right ventricle is pulmonary artery. So this is aorta and this is pulmonary artery. Okay, right. So the openings of this pulmonary and the aorta are actually guarded by semi-lunar valves, which I have mentioned in the previous session. So it is, okay, they are having semi-lunar valves. So here also semilunar walls. So the openings are guarded by these semilunar walls. And I told that this tricuspid and bicuspid wall will not allow the blood to flow back into the auricles, and the blood has no other way that they enter into, okay, from the right ventricle into the pulmonary artery, and from the left ventricle the blood enters into the aorta. We know that this left ventricle has oxygenated blood, whereas the right ventricle, it has deoxygenated blood. The deoxygenated blood enters into the right, the pulmonary artery for oxygenation, and the oxygenated blood from the left ventricle through the aorta, it will be supplied to all parts of the body. Right, this lasts around 0.3 seconds. The next comes is these auricles and ventricles come to a resting point. Okay, so when these ventricles relax, okay, the pressure inside decreases, and this may pull back the blood which entered into the respective blood vessels, okay, back into the ventricles. That is prevented by the semilunar valves. And now the closure of semilunar valves, right, it generates or it produces second heart sound. It produces the second heart sound, and that is dub. So the second heart sound is produced when semilunar valves are closed. The first heart sound is produced when the tricuspid and bicuspid valves are closed. Right. So this is the cardiac cycle. So once again this will be repeated. So once the atrioventricular diastole is completed, and this again begins with the atrial systole, and then to ventricular systole, and then to atrioventricular diastole, and it will be repeating throughout the life of the person. So here one more point, when these two auricles, okay, they are both auricles will contract simultaneously. When they contract simultaneously, the flow of blood into the ventricles is increased, okay, by 30%. Okay, so when auricles contract, the flow of blood into ventricles increases by 30%. So now let's say about the cardiac output. So the cardiac output is the quantity of blood pumped out of the left ventricle, okay, into the dorsal aorta, right at the same time the right ventricle into the pulmonary artery each time. So this is the quantity of blood pumped out of ventricles into their respective blood vessels each time on the contraction. So each time when the ventricles contract, so that quantity of blood that will be pumped into that respective, okay, blood vessel, so that will be from right, okay, ventricle, from the right ventricle into the pulmonary artery, and from the left ventricle into the dorsal aorta. Right. So now let's calculate this cardiac output. So cardiac output is calculated by taking the stroke volume and multiplying with the number of heartbeats per minute. So that is stroke volume into number of heartbeat per minute. Now we need to know what actually is this stroke volume. So here the stroke volume is the quantity of blood pumped out of right one ventricle. The quantity of blood pumped out of one ventricle. So usually both the ventricles will take it around 70 ml. So this stroke volume is around 70 ml. The stroke volume is around 70 ml. Now multiplying these stroke volume into number of beats per minute. So we have seen the number of beats are around 70 to 75. So you can take will take 75, or you can also take an average of 72, no problem in that. Right, so 70 the stroke volume into number of beats, that is 75. I'm taking the maximum 75 beats per minute. This is all in the normal conditions, in a normal person with all the normal conditions. Then you will get around 5250 ml. So this 5250 ml of blood will be pumped out of the ventricles on every minute. So this is around the 5.2 liters. So you can calculate with the 72 also, no problem. So we say that the quantity of blood in a person or in a healthy individual is around 5 liters. So a normal healthy individual has about 5 liters of blood in his body, and that all 5 liters of blood in the body will be pumped by the heart every minute. Right, so that's all about the cardiac output. In the next session we shall see about electrocardiograph, and then we'll also see about regulation of cardiac activity. Thank you.